So think about the person that you are today. Everything from the shape of your nose to your hair color, from your beer belly or your bubble butt. All of this comes about as the result of a lot of fine-tuning at the cellular level. But what exactly controls this machine that makes you, you? All of the instructions for making a body are present on your genetic material, or your DNA. But for the DNA to be useful for your cells, it must first be written into messages that your cells understand. And these messages are aptly called messenger RNAs. Now, while we've known a lot about this process since its discovery over 50 years ago, there's still a lot that we don't understand. In particular, how the cells manage to get rid of their messenger RNAs through particular decay pathways, or the removal of the messenger RNAs. Now, if you think about how we get rid of our waste, we have different recycling bins for paper, glass, or plastic. Cells also have a similar method for getting rid of their messenger RNAs, similar to how we have these different bins. And while you would never think about putting paper in a glass bin, cells also have a similar requirement for having all of these different decay pathways, these different ways to remove their messenger RNA in order to survive. And this is so important that if human children are born lacking even a single messenger RNA decay pathway, the result is a fatal disease. So this is an extremely important process and is worthy of study. And in order to address the question of which messenger RNAs require which decay pathways, I'm actually using plants as a model organism. And this is because plants share all of the same decay components as are present in humans. And as a result, I'm able to raise different strains of plants that lack single pathways of messenger RNA decay. And you can see in this decay pathway 3, this would represent one of my plants that is missing decay pathway 3. And you can see that the messenger RNAs are starting to accumulate. And this is what happens in my experimental organisms. So my goal of my research is to actually take a snapshot of all of the messenger RNAs in these mutants in order to determine the specificity for removal of messenger RNAs. Now, all of these findings from my research will lead to a better understanding of how all of these components lead to the formation of the complex structure, the unique individual that you are today. Thank you.